Hi guys, Miss Francis here. Today we're going to discuss the stages of meiosis. Before we jump into the stages of meiosis, I would like to review some key vocabulary words. So we discussed homologous chromosomes, where homologous chromosomes are two similar chromosomes where one comes from your mom and one comes from your dad. And each one of these chromosomes carry the genes that control the same inherited characteristics. Here you've got a chromosome with one chromatid. In other words, it's unreplicated DNA and it's associated with those histone proteins. Let's say this is from dad. Those green ones come from dad. The green chromosomes come from dad. Now I have a duplicated chromosome, meaning I have gone through the S phase of the cell cycle. I've gone through interphase. Now I have sister chromatids, where sister chromatids have the same DNA sequences as each other. Now let's take a look at mom. Mom is represented by these light green chromosomes. Mom has a chromosome that codes for the same information that's found on this chromosome in dad. So in this chromosome, we are not duplicated yet. So I have one chromatid present. Then this undergoes duplication again during interphase. And now I have my duplicated chromosomes, which are made up of sister chromatids. Now, pay attention to these two. So if I'm looking at this chromosome and this chromosome, those are not sister chromatids. They are chromatids that are on different chromosomes. This chromatid is located on the chromosome that came from dad. This chromatid is located on the chromosome that came from mom. Just a reminder, kinetochores are the proteins at the centromere, which the spindle fiber attaches to. And remember, it's those spindle fibers that enable the chromosomes to be pulled apart. Now, some similarities and differences between homologous chromosomes. Each homologous chromosome, so the one that came from dad and the one that came from mom, regardless if it's replicated or not, has the same length. It has the same base pair length. That's what BP stands for. They have the same centromere positions. They have the same staining pattern. When we do a gel electrophoresis, that DNA would separate into the same staining pattern. Furthermore, the genes are at the same loci, the same location on each chromosome, on the chromosome that came from mom and then the chromosome that came from dad. The differences between homologous chromosomes is that I have one coming from dad, one coming from mom. Furthermore, I might have different alleles present for the genes on each homologous chromosomes, where the alleles represent the different versions of the alleles. This information, when we're talking about heterozygous or homozygous, that we'll discuss when we talk about Mendelian genetics. For now, I'd like you to know that alleles mean different versions for the same gene. Another worksheet to recap homologous chromosomes. They are a pair of chromosomes where I get one from mom and one from dad. Each one of these chromosomes contain the same genes at the same locus or loci, as we saw earlier, which just refers to the location of the gene on that chromosome. The big difference is that they might have different alleles. Different alleles are the versions of the gene. And how do I get those different versions of the genes? It's due to the arrangement of the base pairs. So for example, in one chromosome, my base pairs might be arranged in a different way than in the other chromosome. So my ATs, Cs, and Gs are in a different order. So let's take a look again. Here's my chromosome that has not been replicated that comes from mom. I've got this gene that's going to code for tallness. Now I can have different alleles of the gene that codes for tallness. So here I have the gene that comes from my other parent, where the code for tallness is at the same location, but I could potentially have different versions of that allele. Once I undergo duplication or replication, which occurs during the S phase of interphase, I make a copy of that singular chromosome, and now I have sister chromatids, where the information on this sister chromatid is identical to the information on this sister chromatid, and those chromatids came from one of the parents. So in this case, the orange is the maternal chromosome coming from mom. On the other hand, these purple chromosomes have come from dad. Dad has duplicated or replicated its chromosomes during the S phase of interphase. And again, I have sister chromatids, where each one of these chromatids have the exact genetic information. These are our homologous pairs of chromosomes. In other words, I got one of these pairs of chromosomes from mom and one of the pairs of chromosomes from dad. We also discussed vocabulary words such as diploid and haploid. If I have a diploid cell, it's represented by the notation 2N, meaning there are 2 of every chromosome. In other words, the homologous pairs are present. The function of a diploid cell are those somatic cells. So those are your body cells. So for example, all other cells besides for your sex cells. And if I were to sketch a cell that has diploid number of chromosomes, here my 2N number is 2. These are unreplicated chromosomes. Again, my 2N number is 2, right? I have 1 chromosome, 2 chromosomes, just that my chromosome has been replicated, and now I have sister chromatids. The human diploid number is 46. If I look at my haploid cells, that means there's only 1 of every chromosome. The homologous pairs are absent. Examples of cells with the haploid number would be sex cells, such as your egg and your sperm, which makes sense because those are going to join during fertilization to form a zygote that now has the full set of chromosomes. So if I were to sketch a cell with the haploid number of chromosomes, here my N number is 1, just like in this cell, my N number, or my number of chromosomes, is 1. Here I have an unreplicated chromosome. Here I have a replicated chromosome. In humans, that haploid number is 23.